students to this chapter. And in this module, we shall be having a look at recombinant DNA technology or rDNA technology. In the previous module, we have studied genetic engineering, and this recombinant DNA technology is a type of genetic engineering. So let us move ahead and take a look at the history of rDNA technology or recombinant DNA technology. rDNA technology was proposed by Peter Lopan, who was a graduate student with Amin Dale Kaiser at the Department of Biochemistry at Stanford University. However, their experiments did not succeed, and the real advancement in rDNA technology took place in the year 1972 with the experiments of Herbert Boyer and Stanley Cohen. Now, what these two do? These two scientists, they took a vector plasmid. Yes, recall we have studied about plasmids in 11th standard, and what did they do? They took a vector plasmid. They mixed this vector plasmid with a gene for antibiotic resistance from Salmonella typhimurium. They isolated the gene of antibiotic resistance from Salmonella typhimurium, and they mixed this gene with this selected vector plasmid. Next, what did they do? When they mixed it, they got a new DNA. The DNA which carried the gene for antibiotic resistance. And this new DNA or this plasmid they introduced in E. coli cell for cloning. So Stanley Cohen and Herbert Boyer, they took the vector plasmid, they introduced the gene for antibiotic resistance from Salmonella typhimurium in this vector plasmid, and they got a new DNA which is nothing but the recombinant DNA or the chimeric DNA. And they introduced this new DNA in E. coli cell for cloning. So, recombinant DNA technology, let us have a look at the definition of recombinant DNA technology. So thus recombinant DNA technology is the technique of manipulating the genome of a cell or organism so as to change the phenotype desirably. Okay, so through recombinant DNA technology or through rDNA technology, we can accomplish certain changes in the phenotype by inducing changes in the genotype. So this is nothing but the rDNA technology or recombinant DNA technology, which is a part of genetic engineering. So in this module, we had a look at recombinant DNA technology, and in the next module, we shall be having a look at steps that are involved in rDNA technology. Till then, take care, study well, and bye-bye.